Following Sander's thorough update on the progress we've done so far, I wanted to touch upon the challenges and what options we have to extend gene therapy program for different conditions. So I wanted to just start off by saying by explaining what are the main aims of gene therapy, and that is not necessarily for eye gene therapy, but generally gene therapy in different kinds of organs and tissues in the body. So gene therapy is the right choice when, first of all, there are no efficient alternatives. And usually in inherited conditions, there are few alternatives that would actually treat the symptoms, let alone the underlying cause. One of the main prerequisites to have an efficacious gene therapy application is that some, or in the best case scenario, most of the cells have to be present. So the concept of the gene therapy, as Sandra explained, is we are trying to put back the correct copy of the gene into the affected cell. So by definition, the cell has to be there. What happens if all the cells are not there? We're going to hear later on what options we have. But to have a gene therapy option that would be advantageous to the patient, there have to be some cells present that are affected for us to intervene. The second most important aspect would be to know the genetic cause. Because the big promise of gene therapy is not just to have a one-off treatment, but it's also the promise of tackling the actual cause. We don't want to deal with the symptoms anymore. We really want to deal with the cause. And with inherited disorders, there is a genetic cause. There is the mutations that we heard about, these spelling mistakes, if you like, in the DNA sequence. So if we know it, then we know how to intervene. And I mentioned that this is a best case scenario because there are disorders, and usually the one we hear more about is the age-related macular degeneration. These disorders are complex, which means we don't know exactly if there is a single gene or if there are more genes that cause the disease, if there are other factors. So in cases of complex diseases, what we can do is really keep on looking to find as many more underlying genetic causes for us to have targets. If we don't, what we can do, and that would be our worst case scenario from a gene therapy point of view, would be to try and boost the cell viability of the affected cells, even though we don't know the underlying cause. Again, that would not be as good as actually tackling the genetic cause which is causing the disorder, because again, we end up treating the symptoms. So our main aim is to have cells that are alive, and we know what gene is affected so that we can intervene and correct the effect of the underlying mutation. So what are the challenges when we want to undertake a gene therapy program for a new disorder? First of all, challenges that we have to face generally in gene therapy. When we speak about disease-specific approach, what we mean is a gene-specific approach. Okay? Because when we want to insert a gene back that is missing, it's only this gene that would do the job. So sometimes we hear, for example, RP65 is causing Leber congenital amaurosis type 2. So it's quite specific, the type 2. If another patient has Leber congenital amaurosis, might not think, oh, I think I have the same disorder, so maybe I can use the same gene therapy vector. So it doesn't work like that. Leber congenital amaurosis is a big group of disorders, so you can have a type 10, for example, which is causing mutations in a completely different gene called CEP290. So when we're talking about a disease-specific in this case, it has to be gene-specific. We need to know the gene. We need to work only with that gene. When we start doing our research, apart from knowing the mutation, which is definitely a good start because we know the sequence, we know the sequence of all genes. So if we have identified the gene, and even better, if we have identified the nature of the mutation, then we can start working on it. But once we are done with the design of the piece of DNA that would correct that mutation, and we are ready to put it into our vehicle and into our vector, our viral vector in this case, we need to test it in a model of disease. And the model of the diseases are not always available. So a model of disease can be a cell line that we can use in the laboratory that has the same mutation in that gene, and we can follow that cell line in the dish. We can see how it behaves, how it can develop, what effect it has on that cell line. And then we can take it a step further, and we can have animal models of disease. There are many animal models that they are also naturally occurring. You might have, for example, it happens very often with certain genes, the same gene that affects a human patient, if it has a mutation in the mouse, it actually creates a very similar disorder. And that is very useful in the sense that if you have, for example, a mouse model of a disease that has a similar mutation with the patient and it loses its sight, we can try and treat that mouse and see whether we have an effect. And that is really, really helpful in order to know how efficacious our gene therapy strategy is. Again, we have to make sure that these models we have, whether they are cells or whether they are animal models, that they are also relevant. So the effect of the mutation doesn't always mean you're going to get the same phenotype, the same effect in the viability of the cells, in the way the cells don't function across different species, across different models of disease. So it's not always easy for us to have the correct model of disease. And part of us in the lab, part of our struggle in the lab is not just to design the therapies, but also to design adequate disease models, because these two go together, and we're going to use one to prove efficacy of the other. Another challenge to treat different kinds of gene disorders is the size of the gene of interest. So when we're using a viral vector, we, like Sandra mentioned, we remove the genes of the virus and we place in there the gene of interest, the correct gene that we want to put back in the patient. Now, each virus has a certain size, and this certain size can only fit a certain amount of DNA. So we can't just pack that virus, that viral particle with more DNA than it's able to carry. It's just impossible. So we have a certain limitation into how much sequence, how much DNA, how many DNA letters we can fit in there. So, for example, in the adeno-associated viruses, these AAV viruses we're working with, we have a limit of about 4,000 letters of DNA that the gene has to measure up in order to fit in there. Together with that, we always need to add some additional sequences, some regulatory sequences that will actually tell the cell, give it a direction that, you see the sequence over here that we just inserted? Well, please bring your toolbox in and make a protein out of it. So we need to have all the information in there, not just the gene sequence, but also tell the cell exactly what to do with it. And finally, the other challenge that we have, depending on the disease type and the mutation type, is whether the gene is not functioning or if it's malfunctioning. And there is a big difference between these. So most of the mutations we're currently working have to do with mutations that are causing the gene not to function. So the protein is not being produced. And in this case, you just have to put back a correct copy so that you can replace the production of the missing protein. What seems to be a bit more challenging is to treat a mutation that causes a gene to malfunction. So if the gene is malfunctioning, it means that the protein can actually be there, but the shape of the protein and the actual sequence of the protein is not right. So the protein is doing another function which is not what it's supposed to do. And usually, if a protein is not doing what it's supposed to do, it is toxic. Okay? And in that case, sometimes, no matter how much more of the healthy protein you put back in, that doesn't really help. So just to make a very quick example, for the non-functioning, you have a car, you have a flat tire, you change the tire, everything is fine. If you have a car that the wheels are not aligned, no matter how many tires you change, the most expensive and the best and the brand new ones, it actually won't solve the problem. So in this case, with a malfunctioning, you have to have another way to intervene apart from just replacing the gene. So what can we do to overcome these different kinds of mutations that we will need to deal with to cover as many more diseases as possible? So to start off with the last one mentioned, the malfunction. So we would need to silence the malfunction. If that gene, that mutated gene, is creating a toxic product, we would have to tell the cell to stop that. In order to do that, we have to deliver a different piece of DNA, which actually, instead of telling the gene to produce the cell to produce a protein, it tells the cell, please stop this gene making this product right now. And in many cases, that on its own would be enough to make the disease better. Okay? If you want after that, you might as well deliver some more of the healthy protein, but that's fine, but as long as you remove the toxic product from the cell. And this is something that is currently being studied around the world in different gene therapy labs. It's just because it's more challenging than putting back a copy. Still, it's just in more infancy in terms of delivering more clinical trials. But the process is definitely progressing. The second problem that we might have is that certain genes might be too large to fit into a viral vector. So what happens if your gene is, let's say, larger than 4,000 letters of DNA? There are studies that what they're trying to do is split that gene into two pieces and deliver one half in one viral vector and the other half in a second viral vector. So the idea would be if you go and mix those two viruses together, these two viral suspensions together, you can co-inject them under the retina, and then both viruses would hopefully enter the same cell. And once inside the same cell, they will deliver both their parts inside the center of the cell, the nucleus, and both these pieces would be able to then join together as if they were one long product, and then the cell won't know the difference whether these two pieces came up from one vector or from two. All it sees then is just this gene whole again, and it will be able to drive it, to drive the expression, to make the protein. So this again is being studied, and there are obstacles to overcome. For example, when you use two vectors instead of one, your chances of both of them hitting all the cells, of course, are not the same as putting one message, one gene with one virus. Because there you have one virus that's going to hit all the cells, and you know you have all your message in all the cells. So what we're trying to do is now make this even more efficient in the field so that you can get enough product from both viruses so that you can get even those large genes replaced into the patients. And that would, again, increase the range of gene therapy applications that we'll be able to undertake. One new and very exciting technique which has been developed actually the last couple of years, and it's really one of the beautiful ways to show how molecular biology really never stops to improve the techniques we have. You would think that being able to deliver a correct copy of a gene in the cell would be just enough of what we need in the end. If we can just put back a correct copy, we're good to go. In fact, now we have the tools to take it a step further. Instead of just delivering the gene, we can now go back and edit the actual chromosomes we have. So up to now, we know we have two copies of the genes. Maybe one of them is mutated, maybe both of them are mutated, and well, this is part of the chromosome. We come back and we put a new copy into the cell, and that's good enough. That copy stays somewhere in the cell. It's making its product. We don't care. We're good to go. But actually now we are able to not worry about us delivering the actual new gene, but we can go inside the chromosome and try and correct the mutation within our chromosomes. So that new technique, which is still being developed, would give us the ability to overcome some of the problems we have with the gene replacement therapy. So the size of the product might not be an issue anymore, for example, because it doesn't matter if the gene is 20,000 letters long, when you can specifically home in the mutation in that part of the DNA and just correct the mutation. So you don't worry about having to deliver the whole 20,000 letters. You could just go and correct those 3 letters that have a problem. And again, that has other advantages as well. You correct your own copy means it's going to be produced and make the protein from your own regulatory sequences, these adjacent sequences we talked about that we need to deliver together with a new gene so that we tell the cell to make a protein. We don't have to tell the cell because it has already all the information. We just correct the mistake, and then the cell, it will keep on making as if it never happened. In fact, in theory, if you are able to correct that mutation, there is no way that the cell would know that it ever had this mutation because it's right back into your DNA that you are able to correct it. So this is the big promise of the genome editing, as we call it, technique, and this will definitely dominate the field of molecular biology for the next few years. And will definitely help us to increase the range of the gene therapy we will be able to do in the future. I just wanted to close with what happens if none of this can work. And the answer would be there is always stem cells. And I just want to prepare for the next speaker as well. And with the stem cell studies and research and clinical trials, we can potentially bypass many issues that we might have sometimes with gene therapy. For example, whether cells are not present. And again, with the stem cells, with stem cell research, if you are able to generate healthy cells, you could use that for a range of diseases. Because like we said, gene therapy has to be disease specific, and you have to have some cells there. So if this is not the case, we can always have stem cells to come in. But until then, we keep on advancing our toolbox, our gene therapy toolbox, to be able to edit as many genes as possible to increase the range of the disease availability for our program. And we're going to have plan B and plan A. So whether it's our program getting stronger or even stem cells coming and filling in the holes of where gene therapy cannot help. I'll be glad to take any questions.
Are you saying that there is definitely, and I'm not very bright on this, so forgive me, definitely a link for gene therapy and AMD, or because you can't necessarily link AMD genetically?
No, exactly.
Necessarily, but you may be able to?
No. So what I mentioned AMD as an example of a complex disease. There are others where we don't know if there are any underlying gene mutations. There might be environmental factors. And like with other complex diseases, if you don't know what is causing it, the only thing you can try is just maybe treat the symptoms. Yeah. So that's why AMD was an example on that. But no, it's if in the future there would be some better clarification, if there would be genes identified and all that, then we can talk about it being a candidate. But so far, no.
But you're not working on that?
No.
We're not. No,
it was an example to explain the complex diseases, yeah.
Is there a particular age by which this would work better, or does it matter at what age someone is that this would be carried out?
This, you mean the gene therapy in general?
Yeah,
Yeah. So it depends. It depends on the disease. So different diseases have different progression, which means whether the cells die or not. Some diseases are, as they call, stationary. The cells don't die. So if the cells are there, you might have a larger therapeutic window to go in. And with some others, that you have a smaller window, the earlier, the better. So it depends. It's very much disease specific.
But the actual, the way by which it actually works is going to be the same. So a younger person, it would, let's say it's one particular disease, it would work exactly the same if it was one age or another.
Yes. So to rephrase your question, it would be, would a virus infect young cells better than older ones? And no.
Just a quick one. You use the word protein quite a lot in your talk, and to me, protein means like meat, fish, and eggs. So, what does it mean in your context?
So every gene, which is a sequence of letters of DNA, has one function. Okay, it needs to make a protein. So while meat, fish, and eggs, we identify them as proteins because they are cells that they are very heavy on proteins. So in fact, when you're thinking meat, you're thinking muscle. Okay? When we say we eat meat, we eat muscles. And muscles are just the type of organ, the type of tissue that it's very heavily loaded with proteins. And that's why we say, I eat meat, I take a lot of protein. But in the end, each gene has this function. The DNA is being used by the toolbox of the cell to be transformed into a protein, which is a completely different molecule than the DNA. And each protein has its own function. And we are made largely of proteins, not only the muscle. I mean, really all over the place. We're mainly proteins. So in that sense, I use protein as the final end product because that's the aim of every gene.
Thank you.
Could you elaborate on the editing process a bit? When you have the faulty gene, and how do you correct that one with editing? Could you elaborate on that?
So that's exactly the very interesting part with this new technique. So you need, instead of delivering, let's say, a right copy of a gene that we talked about in gene replacement, in this case, you would have to deliver a different enzyme. So it's a protein that has a specific function. This protein, if you deliver another part together with it, a very small molecule that can actually direct it to find within your whole set of chromosomes, it can specifically find the region where you have a mutation. So you can direct that enzyme to that area. So you would need to deliver that enzyme plus a targeting kind of sequence, different sequence, so that you can go into the piece of the chromosome and then that enzyme is able to remove the mutation and you can correct it.